Have you ever noticed the weaving patterns in clothes? You can see vertical and horizontal arrangements of threads which provide additional support to the cloth and keep the cloth intact. Humans also have a skeletal system which supports all the organs of the body. Likewise, a living cell also has a skeleton which forms a basic framework where all organelles are enclosed. So, let us now focus on the cytoskeleton of the cell. In depth, we can define the cytoskeleton as a filamentous proteinaceous structure present in the cytoplasm. We know that the cell constitutes proteinaceous particles, right? So, let us now check the structures which supports the cell. Microtubules are hollow tube-like structures which provide the basic support to the living cell, and they are made up of the protein tubulin, whereas microfilaments are the structures which are made up of the protein actin and provides the additional support like tensional support to the cell. Let us now check the different significance of the cytoskeleton. It provides mechanical support to the cell. It protects the cell from the external stress. The cytoskeleton maintains the cell's shape. These structures have an effect on the locomotion or the motility of the cell, especially the amoeboid movement. In addition to the cytoskeleton, there are specialized structures for locomotion too. Here, you need to note that there are two kinds of movement. One is the movement of the cell from one place to the another. For example, let's say the movement of sperm with the help of specialized structures. Also, there are some cases where the cell remains static but helps the other particles to move. In the digestion process, there are specialized structures in the digestive system which helps in the movement of food particles from mouth to anus. So now let us focus on those specialized structures whose prime function is the locomotion, and they are cilia and flagella. They are hair-like outgrowths of the plasma membrane. To understand both structures, let us now compare both structures. Basically, cilia are seen in the eukaryotic cell, while the flagella are seen in both prokaryotic and eukaryotic cells. Coming to the size, cilia have a small size when compared to a flagella. The number also differs. Cilia are seen in numerous number, while only a few flagella are seen per cell. Being the structures for locomotion, cilia exhibit fast movements, while the flagella exhibit wavy motion. Now, let us try to describe the formation of cilia and flagella. Cilium and flagellum are formed from the centriole-like structures called the basal bodies. Where are these structures seen? The basal bodies are seen in the cytoplasmic area of the eukaryotic cell where the microtubules are produced. The centrioles of the cell also acts as these basal bodies. So, long protein fibers called microtubules extend from the centrioles and form the spindle fibers which plays a significant role in cell division. Now, let us check the cross section or the ultrastructure of cilia and flagella as if you are viewing this picture from the top. Both cilia and flagella have a similar internal structure. They have a plasma membrane, an axonium which has a central core part, and 9 pair of doublets peripheral microtubules which are radially arranged, and 1 pair of central microtubules. The axonum is a skeleton that gives support to the structure and the ability to bend. As you can see on the screen, the central microtubule are not paired, while the peripheral microtubules are arranged in paired structures. This kind of arrangement is referred to as 9+2 arrangement. The tubules help determining the locations of the nucleus and the other organelles within the cell. Here you can see that the central microtubules are connected by bridges. Next, we will get familiarized with the radial spokes. The central sheath, as you can see in the center of the image, is connected to the peripheral microtubules through these radial spokes. So you can see that each pair of peripheral microtubules have one radial spoke. Thus, 9 radial spokes are visible. Now we know that these peripheral microtubules are in doublets, right? Linkers are the specialized structures that are connected to each other. Thus, we know how the locomotion of the cells takes place, the name of the locomotory organs, and the functioning of the structures. Now it's time to summarize what we have learned.
Manorama Horizon. Learning made simple.